if you think about precision medicine, and precision medicines are these areas like hospitals or pharmaceuticals that will test a patient's DNA, and they're looking to identify a specific mutation that is actionable. They can treat this mutation with a drug. This is a very exciting field, but there is a problem, because many times when you are sequencing patients' DNA, you do not find an actionable mutation. What you find is a mutation that represents a variant of uncertain significance. Can we go back one slide? We're going to get there. Back a slide. So a variant of uncertain significance is a mutation that has never been observed in a patient before. So if you think about it, you can have a mutation or a normal gene, and that normal gene is going to have a normal phenotype. A known mutation might be associated with a specific disease. This is what we're looking for that we can target with a drug. But a variant of uncertain significance represents a mutation that could contribute to disease, but it could just be a passenger mutation, completely harmless to the patient. And when hospital systems that are looking to identify mutations that they can treat with a targeted or precision medicine, variants of uncertain significance represent a problem because you can't do anything with that information. And a significant challenge to these industry settings is that variants of uncertain significance are extremely time-consuming and labor-intensive to validate, and you need a validation process. So to validate a variant of uncertain significance, you can do it in several different model organisms. One way to do it is by using zebrafish. Zebrafish are great. They develop very quickly. In 3 days, a fertilized egg is a free-swimming fish, so you can get information quickly. The eggs are completely transparent, so you can actually witness what's going on and see some of the abnormalities that may be developing. And zebrafish share about 70% of genes with humans. So if there's a gene that is causing the disease in a human, there's probably a gene in the zebrafish. And so what you do is go in with a gene editing tool like CRISPR, and you knock out your target gene, and you look for an abnormality. This is one from our lab. When you knock out a specific gene, you get an abnormal tail formation. Tails should be long or straight. Here they curve over. So we know that when this gene is missing, you get an abnormality that you can observe. The next step is you will do a second experiment where you knock out the gene of interest to generate the abnormality, but then you rescue it with the variant of uncertain or unknown significance. If that mutation is harmful, it causes an abnormality in the patient, you will get the abnormal development. If that mutation is harmless, or an innocuous or passenger mutation, you would get normal development. And so very quickly, within a matter of weeks, you can validate whether that variant of uncertain significance is actionable, it can be treated with the precision drug or not. But again, this is a time-intensive process. So again, this is a problem that industry has. It's very laborious. It takes a lot of man-hours to actually validate just a single variant. And within these settings where they're sequencing lots of genomes of patients, they will find thousands of variants of uncertain significance. Now I want to talk about a university problem. Go back a slide. So in many universities, there is limited capacity for undergraduate research. I come from a teaching institution. At teaching institutions, faculty primarily teach, and so there's less research opportunities. R1s have more research capacity. They have a lot of faculty that are doing research, so students have more opportunities, but oftentimes those opportunities are reserved for advanced students. So students don't tend to do authentic research experiences until later in their academic careers. And at the university I come from, Utah Tech, we're located in southern Utah. One challenge we have in the life sciences industry is there's just not that many industry partners within Washington County. So there's a limited number of internships that we send students to. Internships are a very good way to give students authentic experiences. But again, those students have to travel long distances, oftentimes to the Wasatch Front, to experience those. So you have an industry problem where they need people to validate variants of uncertain significance. You have a university problem where you want to provide authentic research experiences for students. And when we talked to a collaborator of ours, they were explaining this problem of all of these variants that they don't know what to do with. And I was explaining how we have lots of students that we don't know what to do with, and we recognize that there's an opportunity for strong collaboration. And we developed what is known as the Functional Genomics program at Utah Tech. Functional Genomics was funded through a state funding mechanism called the Deep Tech Initiative. Deep Tech partners with university, or provides funding for universities that have industry partners. And it's designed to provide opportunities and training and experience for students to fill this workflow in these critical needs areas in deep tech, or sort of jobs of the future. So we received funding through the Deep Tech Initiative to hire faculty and purchase equipment. We received a significant philanthropic donation to fund student experiences and consumables. And we established the Center for Precision Medicine and Functional Genomics. That is a nexus for collaboration between industry and providing student opportunities. And with this, we created a certificate program. Now, the certificate program is meant to supplement existing degrees. So a biochemistry, or a bioinformatics, or a biology, or even a computer science major can get their degree, but they can get specialized training in the area of functional genomics. We created 4 new courses that train students, and the one thing that I want you to pay special attention to is these classes start with freshmen, moving through the sophomore, and culminate in the junior year. And so the structure of these courses, first students, their freshman year, will take a 2-credit class, so it fits very nicely into their schedules, called Zebrafish Maintenance and Methodologies. And in this class, students learn how to maintain zebrafish, how to feed them, how to take care of them, how to collect the eggs, and most importantly, how to perform microinjections, how to actually hold an embryo with a micropipetter, how to inject it with, we use dye in this class, but they get a skill set where this is going to be helpful in a later course. The next course in the series, students who are now sophomores are assigned a variant that comes from an industry partner. So when we partner with industry, we say, give us your variants, we will validate them for you. So you as a student in this coursework now have a variant from a patient that needs to be validated, and these students will design a CRISPR probe that can target that variant, and they learn how to knock the gene out, and by the end of the semester, they are using zebrafish, and they have successfully knocked the gene out of the zebrafish. And the third course of the semester, now sophomores, will take their variant that they have knocked out, they knock it out of fish, and they're now learning, how do I identify a problem with this fish? They learn a number of techniques like quantitative PCR, Western blot, and in situ hybridization, and they're looking for the abnormality, and in variant validation, this tends to be the biggest bottleneck in the process. It's challenging to actually find out when you knock out a gene, what abnormality do you get? There's behavioral cues, there's actual physical cues that you're trying to look at through a microscope, for example. And then the capstone that these juniors, and they're using the same variant through all these courses, now juniors, they put everything together. They've been assigned a variant, they knock it out, they identify the problem, and in this course, they're trying to rescue it. So they're trying to add the variant of uncertain significance and identify whether or not that variant is actionable or not. And at the end of this course, students will write a report to a physician and outline, yes, when you have this variant or when you identify this variant, you know that it is actionable because we observe a phenotypic difference in zebrafish. This is not the only thing that needs to be done, but this is information that can be used in hospital systems and by pharmaceutical companies to know whether or not a variant of uncertain significance is likely to cause a disease in humans. So I started off my presentation talking about 2 problems, an industry problem of having lots of variants of uncertain significance and for industry to not have the capacity to actually validate them all. And I talked about this challenge of universities, and by integrating this program, we're not only providing students with authentic interesting research projects that they can work on, but we're also validating variants for industry partners. And students, by the end of this course, or by the end of this course series, they are now juniors, and if they want to continue, they could continue to validate variant after variant and potentially have publications, to have significant industry experience. So the program itself at Utah Tech has been an amazing opportunity for students to gain this valuable experience. And students are doing industry-based work in a campus setting, and it's a program that can engage large numbers of students in such a project. So when you look at quotes of students that have gone through the program, they accentuate things like applying knowledge that they're doing in classes, even as freshmen, that the material becomes much more realistic, it becomes much more understandable when you can go to a biology 1610 class and then go to your lab where you're actively doing something that is what you're learning in your normal curriculum. They talk about the hands-on experience, the active learning opportunities, and how it prepares them for future jobs. And this program started 2 years ago. We've already graduated 3 students from the program. 2 of them have gone on to industry settings where they're actually applying the skills they learned. A third has gone on to a graduate program. And I'll also point out that a program like this that is heavily integrated with a strong collaboration with industry has had a tremendous institutional impact, not only for undergraduate students, but also for the institution as a whole. So we are providing transformational experience for students. We're making students more career ready or grad school ready. It has opened the door for grant opportunities that a teaching university, such as Utah Tech, we've written 4 significant grants and plan on submitting 2 more based on this type of program. So it's allowed us to access federal funding that we didn't really have access to previously. It's increased our ability to fundraise, because private donors love this type of experience for students and love the opportunity to provide impact for real-life patients. It has deepened our industry collaborations. We are finalizing a contract now with a local biotech company that is going to pay for our services, so actually pay students to do this work for them. And it's helped faculty recruitment, because we can now recruit faculty with specialized area of expertise to better engage with our students. So for universities that are thinking about engaging and trying to find better ways to engage with not only their students, but with industry partners, finding a program that aligns the goals of the university along with the goals of student engagement will create a high impact. Thank you very much.